Immunology Insights, Basics and Beyond, Chapter 9, B Cells. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we examined the role of natural killer cells in the immune system, focusing on their critical function in fighting viral infections and cancer. Today, we are turning our attention to B cells and the process of antibody production. B cells are a cornerstone of the adaptive immune response, responsible for producing antibodies that specifically target and neutralize pathogens. Understanding B cells and their role in the antibody production is key to appreciate how the immune system builds a long-lasting defense against infections and how vaccines harness this process to protect us from diseases. So let's talk science. What are B cells? Well, B cells or B lymphocytes are a type of white blood cells that play a central role in the adaptive immune system. They originate from the hematopoietic stem cells in the bone marrow and, unlike T cells, they remain in the bone marrow during their maturation. So the B in B cells stands for bone marrow. Each B cell has a unique receptor on its surface known as the B cell receptor or BCR, which is capable of binding to specific antigens. This diversity in BCRs allows the immune system to recognize a vast array of pathogens. The BCRs are essentially membrane-bound antibodies, and when a B cell encounters its specific antigen, it triggers the B cells to become activated. Now let's have a look at B cell activation and differentiation. The activation of B cells occurs when they encounter an antigen that binds to their BCR. However, for full activation, B cells usually require additional signals from helper T cells. This interaction between B cells and helper T cells is crucial as a fail-safe mechanism and also to produce a strong and specific response. Once activated, B cells undergo a process called clonal expansion, where they rapidly divide to produce a population of identical cells, all specific to the same antigen. During this expansion, B cells can differentiate into two main types. First, plasma cells. These are the antibody-producing factories of the immune system. Plasma cells secrete large amounts of antibodies that circulate throughout the body, binding to the antigen that initially triggered the B cell's activation. These antibodies help neutralize pathogens and mark them for the destruction by other components of the immune system. Second, memory B cells. These cells are long-lived and remain in the body after the initial infection has been cleared. Memory B cells do not produce antibodies immediately, but are primed to respond quickly if the same antigen is encountered again in the future. This rapid response is the basis of immune memory and is why we typically only get diseases like measles or chickenpox once. Let's have a look at the structure and function of antibodies. Antibodies, also known as immunoglobulins or Ig, are Y-shaped proteins that play a crucial role in identifying and neutralizing pathogens such as bacteria and viruses. Each antibody has two main regions. First, the variable region. This is the part of the antibody that binds to the antigen. The variable region is highly specific, meaning that each antibody can bind only to a particular antigen. The diversity of variable regions allows the immune system to recognize an almost infinite variety of antigens. Second, we have the constant region. This part of the antibody determines its class or isotype. For example, Ig for immunoglobulin, and then IgG, IgA, IgM, IgE or IgD, and mediates the antibody's interaction with other components of the immune system, such as the complement system or other immune cells. Each antibody class has a different role in the immune response, with some being better at neutralizing toxins, others at opsonization, so the process of marking pathogens for phagocytosis, and still others at activating the complement system. Antibodies can neutralize pathogens in several ways. They can block the binding sites on a virus or bacterium, preventing it from entering host cells. They can also agglutinate or clump together pathogens, making them easier targets for phagocytosis. Additionally, antibodies can activate the complement system, which leads to the lysis of pathogens. We will go more into detail of the molecular aspects of antibodies and their functions in a later chapter of this podcast. Also, T helper cells play a role in antibody production. T helper cells are essential partners in the activation and differentiation of B cells. When a helper T cell recognizes an antigen presented by an antigen-presenting cell, or APC, such as dendritic cells, it becomes activated and secretes cytokines. These cytokines provide the necessary signals to B cells, promoting their proliferation and differentiation into plasma cells and memory B cells. By the way, B cells can also function as APCs, complementary to dendritic cells, to fine-tune and direct the immune response. The interaction between B cells and helper T cells occurs primarily in the germinal centers of lymphoid organs, such as the spleen and the lymph nodes. In these germinal centers, B cells undergo a process called somatic hypermutation, which introduces mutations into the variable regions of the antibody gene. This process allows the immune system to fine-tune the specificity and affinity of antibodies for their target antigens. In a later chapter of this podcast, we will go more into detail on T cells and their interaction with B cells. As the immune response progresses, B cells can undergo class switching, where they change the isotype of the antibody they produce without altering the specificity for the antigen. For example, a B cell might initially produce IgM antibodies, but as the immune response matures, it could switch to producing IgG antibodies, which are more effective in certain types of immune responses. Affinity maturation is another critical process that occurs in germinal centers. B cells that produce antibodies with higher affinity for the antigen receive a survival signal, allowing them to dominate the response. This selection process ensures that the antibodies produced later in the immune response are more effective at neutralizing the pathogen. Let's have a closer look at memory B cells and long-term immunity. One of the most remarkable features of B cells is their ability to create a long-lasting immune memory. Memory B cells persist in the body for years, sometimes even for a lifetime, and can respond rapidly upon re-exposure to the same antigen. This is the principle behind vaccination, where exposure to a harmless form of the pathogen's antigen can prime the immune system to respond quickly and effectively if the real pathogen is encountered later. Memory B cells are highly specific and can undergo further affinity maturation upon re-exposure to the antigen. This ensures that the immune response is not only faster, but also more effective with each subsequent exposure. Antibodies are remarkable, and they can also be used in therapeutic applications. The ability of B cells to produce highly specific antibodies has been harnessed in various therapeutic applications. Monoclonal antibodies, which are antibodies produced from a single clone of B cells, are used to treat a range of conditions, including B cell-related cancers like lymphomas and multiple myelomas. Monoclonal antibodies can be engineered to target specific antigens on cancer cells, marking them for destruction by the immune system. They can also block inflammatory pathways in autoimmune diseases or neutralize toxins and pathogens in infectious diseases. The development of monoclonal antibodies has revolutionized modern medicine, providing targeted treatments that are both effective and specific. While B cells are essential for immunity, their malfunction can lead to significant health problems. Among the most notable While B cells are essential for immunity, their malfunction can lead to significant health problems. Among the most notable are B cell cancers, such as multiple myeloma and B cell lymphomas. These conditions arise when B cells grow uncontrolled. While B cells are essential for immunity, their malfunction can lead to significant health problems. Among the most notable are B cell cancers, such as multiple myeloma and B cell lymphomas. These conditions arise when B cells grow uncontrolled due to mutations or dysregulation in their normal development pathways. The multiple myeloma affects plasma cells, a specialized type of B cells. It leads to the overproduction of abnormal antibodies, which can damage bones, impair kidney function, and suppress Multiple myeloma affects the aforementioned plasma cells. It leads to the overproduction of abnormal antibodies which can damage bones, impair kidney function, and suppress normal immune responses. B-cell lymphomas, such as the Hodgkin and non-Hodgkin lymphomas, are cancers that originate in lymphoid tissues where B-cells reside. These cancers can result in enlarged lymph nodes, immune suppression, and systemic symptoms like fever and weight loss. Interestingly, therapies targeting these diseases often exploit the same mechanisms that make B cells such potent immune agents. For instance, monoclonal antibody therapies are designed to selectively target cancerous B cells, leveraging our understanding of B cell biology to treat these malignancies effectively. To conclude, B cells and the antibodies they produce are central to the adaptive immune response. Their ability to recognize a vast array of antigens, produce highly specific antibodies, and generate long-lasting memory makes them indispensable in our body's defense against pathogens. In our next chapter, we will explore T cells, another critical component of the adaptive immune system. We will dive into the roles of helper, cytotoxic, and regulatory T cells and how they coordinate to protect the body from infections and to maintain immune balance. Until then, remember that the antibodies circulating in your body right now are a testament to the incredible power and precision of the immune system. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.